the knowledge, the scientific foundations about the glycemic index are possibly more solid than anything else in nutrition. And now I'm going to show you why. All right, so when we talk about glycemic index and use the abbreviation, GI, we're not talking about the gastrointestinal tract. We're talking about the glycemic index. And in particular, the glycemic index is relevant because in medicine, postprandial glycemia is a risk factor, increasingly acknowledged as a really important risk factor, not just for people with diabetes, but for people with pre-diabetes and even normal glucose levels, particularly in pregnancy. So postprandial glycemia is relevant to the prevention and management of type 2 diabetes, gestational diabetes, GDM, cardiovascular disease, obesity, and also, perhaps the evidence is less mature, but in type 1 diabetes, it's relevant, in polycystic ovarian syndrome, which they now believe affects as many as 1 in 5 women, which makes it the most common endocrine disease of all, that it affects pregnancy outcomes, adverse outcomes like very large babies, and therefore child obesity, that it has applications in sport, and, interestingly, in mental performance as well, cognition. So, just about relevant to everybody on the planet. Okay, why is postprandial glycemia relevant? Well, we know from particularly research over the last 5 years, and I'm sorry you haven't got this slide, don't go looking for it because I added it at about 4 o'clock, all right? I realized that there was a gap there. I needed to tell you why high glucose levels are dangerous. You know they're dangerous for people with diabetes and pre-diabetes, but you know the high normal glucose levels are now considered to be a risk factor. So, glycemia produces insulinemia, and it goes back in the other direction. Even just physiologically raised insulin levels produce insulin resistance. And together, those two things, sometimes it's hard to separate which is the primary cause, but it produces oxidative stress in the cells. Lots of free radicals are generated. These highly reactive oxygen molecules do harm, particularly inside the mitochondria. This produces inflammatory responses, cytokines, and transcription factors that produce a whole lot of downstream effects, which are interpreted by the cell as inflammation. And this in turn affects endothelial dysfunction. So, endothelial layer of cells lining the blood vessels, these are early signs of atherosclerosis, that those blood vessels are not responding the way they should. It also affects the degree to which blood coagulation takes place, that a thrombus forms, or a thrombus forms and then dissolves, all under the influence of oxidative stress. And beta cells are the glucose sensors in the body. They're the most sensitive cells in the body, and so they should be. But if you work them really, really hard, and their mitochondria are overworked, then their mitochondria disappear, and then the beta cells themselves start to die. So, that, of course, once they start to die, you see the beginnings of type 2 diabetes. And finally, relevant to everybody, glucose is a molecule that sticks to proteins. And the higher the concentration of glucose, the more it sticks. So you all know that glycated hemoglobin is a measure of average blood glucose levels in people with diabetes. Well, glycated hemoglobin is a hemoglobin protein molecule with glucose stuck to it. All right? But every protein in the body is susceptible to this glycation process. And you know what? The proteins, once they're glycated, they're irreversible. And they don't do a good job anymore. Glycated proteins are dysfunctional themselves. And so those wrinkles, everything about our body that ages is to do with advanced glycated end products, so wrinkles. All right, so let's get to carbohydrates. Carbohydrates are the only food constituents that directly raise glucose levels. If I give you some olive oil to drink or I give you some fillet steak to eat, your glucose levels will stay as a flat line, okay? So foods like bread and pasta are very high in carbohydrates. But you know, the carbohydrates are not identical. If you look at them under the microscope, there are important differences. And you know, your mouth can detect those differences. You think about it. You could put a morsel of fluffy white bread in your mouth, and within 2 minutes it would sort of dissolve in your mouth because your amylase in your saliva is attacking the starch. But you put a piece of pasta in your mouth for 2 minutes. At the end of the 2 minutes, the shape of the pasta is still detectable. Spirelli pasta or penny pasta. And that's because the degree to which the starch in those foods is being broken down differs. All right, so gram for gram of carbohydrate, or if you like, weight for weight, or ounce for ounce, carbohydrates vary in their glycemic potency, or glycemic impact, or glycemic punch. And glycemic is just a medical word for blood glucose or blood sugar. So, in layman's terms, we can say some carbohydrates are gushers because they're digested and absorbed very quickly, and they gush, the glucose gushes into the bloodstream. On the other hand, there are some carbohydrates which are digested and absorbed more slowly, and those are called trickles. And you know, in theory, you could have a carbohydrate that trickles so slowly into the bloodstream that you've got a flat line. All right, so the glycemic index is just a way of expressing these differences between carbohydrates in different foods. And it takes the foods as they're normally eaten. All right? You have to have a food that can be capable of eaten, that's eaten. You have to have a carbohydrate food that has sufficient amount of carbohydrate that it's called a carbohydrate food. You can't test the GI of tuna or eggs or meat or chicken, only foods that contain carbohydrate. And we're comparing them gram for gram. So it's just a tool. And when it comes to glycemic index methodology, I don't think there is anything in nutrition as well assessed or measured as the glycemic index. So, just to show you why, just remember we're comparing carbohydrates gram for gram. So, it's like comparing protein quality, the protein quality in chicken versus egg versus lentils. It's the same thing. We're comparing them by looking at the area under the curve over 2 hours. The area under the curve is just a nice statistical way of assessing the average glucose level across 2 hours. We express it relative to a reference food, and the reference food is usually 50 grams of glucose. But you know, it could be 25 grams of glucose, it could be 20 grams of glucose. All that matters is that there's the same amount of carbohydrate in the test food and the reference food. Because remember, you're comparing them gram for gram of carbohydrate. And because we want to standardize glycemic index testing as much as possible, we usually use 50 grams unless that's too big a load of food and we come down to 25. So the glycemic index of the reference food by definition is 100, and to assess the glycemic response of any one test food, be it a new bread or a new breakfast cereal, we need at least 10 subjects. All right? So in our lab, because we've tested the reference food 3 times on 3 separate occasions in every single individual, and we do that once a month so that we've got a running average of their glucose tolerance status, and because we test the food once and we test it over 2 hours and we take 8 blood samples and we do them in duplicate, we've actually got 640 data points to generate the glycemic index of one food. Pretty amazing, isn't it? All right? And then they talk about the measurement of fibre. You know, if I measure the fibre